אתה יודע מה להגיד?
תורת אצל קריס. תורת אצל מסך.
סטריט קמפוס סמילר.
זה בשידור עכשיו.
כן? זה טוב, תצנזרו.
שומעים אותנו?
אתה זוכר איפה רוצים סטופ סטרימינג?
לא, בוא תראה.
Now they hear us in Eilat. Everything is safe. כאן?
אוקיי.
סטופ סטרימינג.
Now that they saw my face, they know that they've already done the same.
Now they're going to back to lunch.
To the beach.
רגע, maybe Shai is listening now?
Okay,
So you need a pointer?
I'll tell you in a second. First, I will try. So it's not connected with normal source. Should I compute it?
Yes. So, okay, so, wait. Okay, right?
Let's see. Okay.
רגע, עכשיו.
אני עושה הרצאה הקדמה לעצמה.
אם אתה רוצה להציג אותנו, זה יהיה סבבה. כן. So you need a pointer or not?
Yes, I need a pointer. Sorry, it doesn't work. Do you know what this is?
What?
Do you want me to try and get my buttons?
אין סוללה?
Yay! Let's try it. Let's try with this one. Oh yeah, is it the big one?
You need a big screen in front of you?
לא, מה אתה עושה?
No, it's okay, I will point anyway here. Want to try this one? But I think these ones are too big, no?
Triple A, what you have?
Let's try this one. Mine didn't work, maybe.
No, it should.
If my batteries were a bit...
No?
Okay.
I'm just going to bring another one.
Let's try this one. Or maybe put this one here.
This one? Ah, okay, it's better.
Yeah, but now this is, you got this one connected. So just test yours. In the meantime, I'll go bring another pointer.
Yeah.
Okay, it works.
I don't know. Looks like it's working. Yeah.
Okay. Cool.
Cool.
You can keep the batteries.
Can I?
Present.
תודה רבה.
Live on the YouTube.
What?
I see you live on YouTube.
Okay, hello everybody. I'm happy to present Flora. Flora did a PhD in Paris under the supervision of Chris Bowler. Now she's on her postdoc with Professor Asaf Vardi in the Weizmann Institute. She will talk on diatom interaction in the oceans. Flora, the floor is yours.
Thank you. So yeah, first of all, hi everyone, and hi also to the people in Elat that saw me 6 months ago. I think they're live streaming. So yeah, as Nadav told you, I'm here today to talk about what I did during my PhD when I was in Paris with Chris. And just to give you a brief background, I'm not initially trained as a marine microbiologist. I actually started as an environmental engineer, agricultural engineering, but I actually quickly moved on to marine biology that I was quite lucky to discover in Japan and French Polynesia. And then following my master's degree, I decided to do a rotation in Chris's lab in Paris, where I discovered the Tara Oceans project that I'll present. And now today, I'm in Israel since 1 year in Weizmann, and it's a lot of fun. So thanks for coming to this presentation. So just a brief presentation of like the outline of the presentation. I'll start with the introduction on Tara Oceans, on diatoms, because I don't think everyone here is familiar with unicellular microeukaryotes, and then move on to the two major parts of my PhD. One of them is more, let's say, computational based with a very large scale inference of community interactions, and the second part is a bit more tailored towards a specific type of interaction that we study in depth and that we try to characterize morphologically, genetically, and in terms of environment. So this part is a bit more, let's say, conceptual and bioinformatics based. This one is a bit more experimental, so you can choose on which one to focus if you prefer one of them, and then ending up with the perspectives. So as you know, the ocean comprises 71% of the Earth's surface. We know today that the ocean plays a key role in regulating climate, that it's also serving as a huge resource in terms of energy, of food, of pharmaceutical compounds. And one of the things that is still surprising with the fact that it's such a big continuum on our planet is that we estimate that we've only explored 5% of the ocean. One cool fact is that more people walked on the moon than touched the deepest parts of the Earth, and that we have more publications on space than on the ocean. So it's like right next to us, and despite that, we are still looking towards 300,000 kilometers from here. And among the things we actually still don't know quite well about the ocean is what's living in it. So we all obviously know whales, dolphins, manta rays, but in reality, 98% of what lives in the ocean, 98% of the ocean's biomass is comprised of microorganisms that are invisible to the naked eye. We call them plankton. So plankton, it comes from the Greek eros. It means errant, so it can't swim against the current. So the only definition of plankton, of microorganisms that holds, it's basically organisms that float in the water column and cannot swim against the current. So to give you orders of magnitude, typically in 1 liter of seawater, you can have up to 100 metazoans that will be small crustaceans that we call copepods. You can have up to 10 million protists. So protists are unicellular microeukaryotes, up to 1 billion prokaryotes, bacteria, and 10 billion viruses. So, okay, they are abundant in 1 liter of seawater, but are they really important? So, in fact, most of the services that are provided by the ocean that we know today are operated by those marine microbes. Typically, photosynthetic organisms in the ocean will fix atmospheric CO2 and they will use it to produce oxygen. Oxygen production, we know today that the phytoplankton in the ocean are responsible for half of the primary production that occurs each year. We also know that all this plankton is actually the basis of the marine food web, so those microorganisms will be fed upon by fish larvae, themselves fed upon by higher organisms up to the top predators, and of course we rely on all this food chain for fishing resource, and also many of those organisms are also good indicators of ecosystem health. So you can see that within the plankton, within the planktonic ecosystem, you are basically covering the whole realm of living organisms, from bacteria, eukaryotes, viruses, and higher organisms. So beyond this catalog of what's actually in the ocean, you can imagine that being so densely populated in 1 liter of seawater, they don't live in isolation, and like many of us, we actually interact. And we have a diversity of microbial interactions like competition, symbiosis, predation that actually occur between these different organisms. And what this type of study actually brings like knowledge is, for example, understanding species coevolution. So do species in the ocean, microorganisms in the ocean evolve by competition or by predation or by symbiosis? You can also, by studying those microbial interactions, get insight on what are keystone organisms. So organisms that, you know, if they disappear, the whole food chain, for example, will collapse. So understanding keystone organism is something quite important regarding microbial interactions. And finally, also, something that we don't always imagine is the impact that those microbial interactions will have on very large scales and on very large nutrient cycles. And one very emblematic case is that of the carbon cycle. So for a long time in the ocean, we thought that there was this basal trophic level of phytoplankton, photosynthetic organisms, that basically fix atmospheric CO2, and that these phytoplankton were basically eaten by zooplankton, higher trophic level, small crustaceans, and that the carbon was kind of cycling here and that it was vertically transferred up the food chain. That was the, let's say, basic thing we thought about a carbon cycle related to biology in the ocean. And then all this view was basically challenged when we discovered those heterotrophic bacteria right here. Those heterotrophic bacteria are actually capable of remineralizing parts of the organic matter from the phytoplankton, so it's kind of conducting the carbon away from the zooplankton. And once the carbon is in this pool of heterotrophic bacteria, it can either be eaten again by zooplankton, so you're linking back to the initial cycle, or it can also fuel those heterotrophic bacteria, can basically sink and fuel the microbial carbon pump, trapping the carbon in the deep sea floor over a geological timescale. So you're moving from one view of, this is just the way it works, to adding a new player that interacts with those phytoplankton, with those zooplankton, and basically kind of rewiring the direction in which the carbon will go. So bacteria are not the only players. We also know now that viral interactions can play a role. The way viruses in the ocean will induce demise in phytoplankton will also change the way carbon is redirected. So all in all, those interactions also help us get understanding of what's happening at large scale. And for a long time, when we discovered heterotrophic bacteria, people were focusing a lot on the study of microbes in the ocean, but on prokaryotes. And this left a huge gap to study protists that are pretty ubiquitous, but really overlooked. So protists are eukaryotes, unicellular eukaryotes. Some of them can be phototrophs, some of them can be heterotrophs. They can live in single cell, but also form colonies. And if you look at this, this is eukaryotic tree of life. You can see that protists are basically inhabiting every branch of the tree of life. They actually comprise the majority of the diversity of the eukaryotic tree of life. And here it's just showing the divergence between animals and sponges, okay? This is how close they are in the tree of life, and this is how diverse the kingdom of protists actually is. So they are actually much more abundant and diverse than what we initially thought. So, and we don't really know actually much about the ecology of protists. And so during my PhD, I focused particularly on diatoms that are right here. Diatoms are also unicellular photosynthetic organisms. They are actually pretty cool. They are unicellular organisms that are enveloped in a silica cell wall. So it's basically two blocks of silica that are combined together with the cell in the middle. And you can see that they come in very different shapes and sizes. And these are drawings that come from the late 19th century, but already at that time we had good microscopes to be able to see how beautiful they were. So what we know from fossil records is that diatoms, those unicellular photosynthetic organisms, the first fossil records go back to 250 million years ago. That since then we had 4 major episodes of divergence that led to 4 major lineages: the centrics, multipolar centrics, the one with more weird shapes, raphid pinnates that are the elongated one, and araphid pinnates that are also elongated, but they don't have a raph in the middle. We know also that diatoms are the product of a secondary endosymbiosis, meaning more complex actually, meaning that you have the successive engulfment of a cell by another free living organism that created, for example, first photosynthetic organisms. We also know that diatoms are pretty ubiquitous in any aquatic environment. They are found in open water in the middle of the ocean, in polar and tropical waters, in fresh water also. You go here in any of the lake, you take a scoop of water, you will see diatoms. They also inhabit soil, snow, glacial ice. And in terms of ecological importance, we know that among all the photosynthetic activity in the ocean, phytoplankton, diatoms are approximately responsible for 40% of it. So even there are quite big players in primary production in the ocean. So from what we know in the literature about diatom interactions is that, again, because they are ubiquitous, pretty well, let's say, studied in the past, we also know that diatoms are quite important in the planktonic community itself. Diatoms establish, we know it from the literature, quite some interactions. Typically copepods that are small crustaceans feed on diatom, and it's known that diatoms in return are actually capable of producing aldehydes that will decrease egg hatching success in those copepods. We also know that dinoflagellates, which are another class of protists, compete with diatoms for nutrients, and there is a lot of research done on allelopathy, which will basically be the competition between those two species. We have typically parasitic interactions. This is a chytrid, and we know that it infects diatoms, and the diatoms in return develop what we call a temperature refuge. So they establish an ecological niche in which the diatom can live, but not the parasite. So this is how they will escape the parasite. And some interactions will involve bacteria, because all of these are eukaryotes. Typically mutualistic interactions. So here you see it's a nitrogen-fixing cyanobacteria that lives within a diatom. So the diazotrophic bacteria will basically provide nitrogen to the diatom, and the diatom in return will serve as a shelter. Not all prokaryotic interactions end well. This one is an algicidal interaction where the bacteria will basically kill the diatom. And there's a whole array of interactions that we don't really know what's happening. We don't have any explanation. Typically, this is a chain-forming diatom. You can see the different blocks, and it has those small ciliates, vorticella, that are attached to it, and we don't really know what's happening, and there's no real mechanistic understanding. So the thing with all those interactions is that most of them have been studied in the lab. Our understanding, which is crucial, is based on cross-culturing or co-culturing of two, in best of cases, 3 different species. But at a, let's say, larger scale level, there are many things that we still don't really know. For example, we don't really know how frequent all of these associations are in the ocean. Most of the time, these are actually associations that we manage to grow in the lab, but are they really ecologically relevant? We don't really know in what environmental conditions, they really occur. And again, something that I didn't mention is that from previous literature, we know that diatoms are considered as good competitors in the ocean. They can produce toxic oxylipins, they can produce aldehydes, they can form chain. They are considered to be good competitors. But again, it's a bit like a third empirical feeling, but the question was, can we actually translate this competitive reputation of diatoms in the way they are actually distributed in the ocean with respect to other organisms? And after, how do these interactions with diatoms structure the whole plankton community as a whole? So obviously these are pretty broad and open questions that require quite a data set that would kind of cover both the whole realm of potential interactions and both various spatial, let's say, environmental conditions. And this is how we get to the Tara. The Tara is actually, the genesis of the Tara Oceans project is pretty cool. It starts with this book, The Voyage of the Beagle, from Charles Darwin, what he wrote when he went on his expedition. So there is a long history of expedition to travel in the ocean. And obviously, this book kind of led Charles Darwin to formulate all the theory of evolution by natural selection. So exploration is good to, let's say, build new knowledge. And it's combined with this guy, who is called Eric Carcenti. So Eric Carcenti was a cell biologist in Heidelberg in EMBL. And he's actually also a sailor. And he read this book in the year 2000, and he decided to kind of conduct a new expedition using state-of-the-art tools of new sequencing, like every new, let's say every new tool we had under possession, to this time not look at whales and dolphins and birds, but to look at microbes. Back in year 2000, it was really the start of high throughput microbiology. We were discovering microbes everywhere, in our body, in the city, in the soil, so no reason not to do it in the ocean, and this is how basically the genesis of Tara started. So Tara Oceans is a 4 years expedition, 2009 to 2013, that was conducted on this boat. It's a schooner, so it's a not very typical type of expedition boat. It's not very practical to do science, but it's been optimized to do so. And for 3 years, the boat basically traveled in over 230 different stations, going from eutrophic to oligotrophic places, so rich in nutrients and poor in nutrients, equatorial, polar regions, covering also different types of biomes, oxygen minimum zones, very high cold regions. So the idea was to try and cover as many, let's say, ocean provinces as possible. It was a big consortium. It's over 50 different teams of very different disciplines, and the idea is to look at viruses, bacteria, protists, but also metazoans. So this involves sampling at different size fractions, sampling at different depths and the surface and the deep chlorophyll maximum. And especially what motivated the consortium at that time was 3 major questions. To basically be able to chart the microbiodiversity that was in the ocean, because we didn't really know what's the state of the art of microbes in the ocean, to understand what were the environmental factors that drove this diversity and this abundance of microbes in the ocean, and to understand how all this community was structured at large spatial scale. And the idea was also not to produce only data for the consortium, but to make this whole data publicly available. So anything that comes out of Tara Oceans and out of any Tara expedition is basically completely freely usable by everyone. So each of the samples was basically processed to each of the stations, had a set of samples. Some of them were for high throughput sequencing. So you have to imagine that in each station, for each size fraction, we had first meta-barcoding. This is a way to chart species diversity in one specific body of water. Metagenomics, which give access to gene diversity. Metatranscriptomics, which give access to gene expression, and each time it's for the whole community. Then some of the samples were also processed to do high throughput imaging, confocal microscopy, which gives access to structural, like the interior of your cell, where you can basically stain the nucleus, the chloroplast, the membrane, see the shape. So it's not only about genomics, it's also about morphology. And finally, each of the stations, because our idea was not to look only at the biology, but also at the chemistry and the physics, was to characterize all of those stations in terms of environmental conditions, to try and see the interplay between the biotic part and the abiotic part. So out of all of this, there are 5, there's a special issue that came out 3 years ago, which already releases 40 million, like a big catalog of bacterial genes, as well as a big repertoire of eukaryotic species. It's a lot of data, but it's only 1/4 of the station, so it's based on 68 stations from the whole Tara database. There are over 220 in total, so there's many more things that are to come. But the idea is that right now we also have a catalog of expressed genes. So if one of you is working on his favorite gene, you can basically go and see how it's expressed in the ocean. You can go and see in which species it's present. You can go and see in what environmental condition it's upregulated. So the whole idea is to make it freely available to the scientific community. So with that, I can move to the first part, which is basically trying to understand global patterns of diatom interactions in the open ocean. So ecology has a pretty long history for studying ecological interactions, biotic interactions in the form of a network. It's what we call ecological networks. An ecological network is a representation of biotic interactions. So what I call biotic interaction is typically symbiosis, competition, predation. And you represent it under a form of a graph where each node is a species, and the species are connected by a small link, which is basically your interaction. And your interaction is characterized based on the relatively positive, negative, or neutral effect organisms have on each other. So traditionally, in ecology, people have been building separately 3 types of network. food webs, so typically predator-prey, host parasites, and mutualistic, typically plant-pollinator networks. So they separated them in 3 different classes. Most of the time it was based on observed interaction, so scientists sitting in the field looking at which bee is pollinating which flower, mostly macroorganisms, so things you can see with the eye, and mostly terrestrial. What this body of literature, so this is one of the first actual networks that was built in agricultural study back in 1880s, so pretty old. And what this visualization of interactions between species in the form of a graph opened the door to using graph theory, network topology, to describe those graphs. And what you can do once you try to extract mathematical metrics from those graphs is to get insight on how robust your community is, how robust are your interactions, who are the species that are really key and central in the community structure, how specific are the interactions. Typically, is one bee pollinating all of the different flowers, or is it more specific, etc., etc. And now there's very nice work looking at how climate change will basically affect the network of interactions between species. So the whole idea was, how do we adapt this to marine microbes, or to microbes whatsoever? So with the arrival of computational power, next generation sequencing also, it became possible to do it with co-occurrence networks. A co-occurrence network is basically a huge map that will rebuild who sticks with who. So I'll go quickly into the details of the method, but I will be very brief to show you how it's been built for Tara. We started with environmental samples, okay, that we collected in different places of the world, and from those bodies, from this biomass of different waters, we get both the information of the diversity of eukaryotes, of prokaryotes, but also of viruses. And from this, you can build a huge matrix that tells you what is the abundance of your organism in each of your sample. And from there, once you have this abundance table, you can, which is here, well, you have relative abundance or presence-absence, we don't really care, with your different species saying if it's present in sample 1, sample 2, sample 3, you do pairwise correlation. So each time you try to correlate the abundance of one of your organism with the other organism. So for example, A is not correlated to B, but it's more correlated to C. You can use different type of metrics. There are tons of ways to statistically validate what you are actually doing. And in the end, what you obtain is a network output where your species are the nodes, and they are connected by different edges that are either green if, for example, here, D and is it B, I don't see, are positively associated in the samples, meaning that if you see one species, you often see the other one, or on the contrary, if you see one species, you never see the other one. So correlation-based. So one obvious criticism when you start from here is that, but two species can be in the same sample just because they share the same environmental niche, right? They might not even interact biologically. Completely true. So one of the techniques that was developed by Caroline Faust that is behind this whole method is to control for the environmental factor. To be able to say, species A and B are in the same sample, but just because of, I don't know, phosphate or nitrate. So you can actually control for that, and we actually have that data in this big network where you see what drives the abundance, like the correlation between two species, whether or not it's based on just a certain environmental factor or actually the presence of the other species. So this was what was done for Tara. So this big unexplainable hairpin ball is what we call the Tara interactome. It comprises both viruses, bacteria, eukaryotes. So again, each node is a species, and they are all connected by those different links. And just to make sure on the definitions before I go on, what I call an interaction is basically when the relative abundance of two species is related to one another and it's controlled for the effect of the environment. When I talk about co-presence, it's that two organisms are often abundant in the same samples. When I talk about exclusions, it means that two organisms are rarely abundant in the same sample. I'm not talking about interactions, biotic interactions at that level. And here my goal was basically to extract everything that concerned diatoms and to try and understand how the diatom interactome was structured at the level of the ocean. So the first question was to say, okay, we have this big interactome with environmental factors. So what proportion of the diatom interactions are actually driven by abiotic parameters? So the way to do it is, here you have all the different environmental parameters that we controlled for. I took off the legend because it wasn't really clear, but it's phosphate, nitrate, temperature, silica. We have a whole bunch. And here are the number of interactions that involve a diatom but that are driven by a third environmental parameter. So typically, if this was a temperature, it's a log scale. If this was temperature, the total number of interactions that are driven by temperature is here, and you have the breakdown between whether it's positive or negative. So all of these are actually environmental parameters, and the big one here is actually none. It means that the interaction, the correlation between my two species is actually driven by nothing else than the abundance of the two species. It's not driven by temperature, it's not driven by nitrate, it's not driven by silica. The two organisms are correlated just because of the abundance of each other. So this meant that basically biotic factors, the presence of other species, was a major driver rather than nutrients. In 87% of all my interactions, there was actually no effect of the environment on structuring this interaction. So the second actually very...
So what you're saying actually is that those people talking about nutrients and nutrients and nutrients
exactly.
are talking nonsense.
Mm-hmm. It's not Stuyot. This is a word that I learned. But it's true that in marine microbiology, there's been a bit of focus on what we call top-down or bottom-up factors. So typically, bottom-up will be abiotic factors, so nutrients, so the effect of nutrients on the abundance and the diversity of species. But more and more, we are starting to understand that there are what we call top-down factors that, like interactions, typically biotic interactions that also shape the distribution and the abundance of species. So yeah, I think there's been a lot of focus on the impact of nutrients, but also evidence from Tara, everything that we have points to the direction of the interactions between species is a driver of evolution, is a driver of abundance, etc.
So, if I may ask a question.
Yeah, of course.
I saw a documentary regarding diatoms when in the South Pole, when you have like a big Arctic
current.
It melts down, then it creates a bloom of diatoms.
Mm-hmm.
So how can you explain that?
I'm not saying that nutrients don't have an effect, but I also think you need a combination of high nutrients but also low predator pressure. Because if you have a lot of predators, you can have a lot of nutrients, but you will still have So I'm not saying that one is better than the other. I'm just saying it's more complex. It's a combination of both. If you want a diatom bloom, you also need to decrease the predator pressure, for sure.
Just a remark to what Alan said. The 87% are related to interactions, not to the abundance or misabundance of species.
No, what the 87% are saying is that even if you, like the best predictor of the abundance of species A is species B.
It's not the interactions, the appearance.
It's correlation.
What is the size?
So the average size of a diatom here is between 20 to 180 micron. So it's quite small. Copepod is a bit higher. We're in the order of 2 millimeters. But yeah, everything here is below 2,000 microns. So we're really in the micron scale. So another very interesting fact when you just look at as basic measures of correlations, positive, negative, is that most of the total interactome has around 70,000 positive edges, okay, and it has around 23,000 exclusions on average. And when you look at the relative contribution of diatoms to each of those processes, you can see that on average, diatoms correspond to around 10% of the total exclusions, only 3% of all the correlations, and that on top of that, within the diatoms themselves, they actually tend to exclude more than they actually co-occur. So if you look, they are basically antisocial. There's a nice paper from 2016 where this guy actually tries to put biological meaning on those correlation networks, and this is a property that he defines as segregators, meaning that on average, those diatoms, they tend to actually exclude other organisms. So there are some organisms that don't really care, that will be more or less with every potential plankton organism, and there are some organisms that are much more segregative, and diatoms are part of them. What is also quite interesting is when you focus on this pattern of exclusion actually caught our mind because it fitted well in the concept of diatoms being good competitors, right? You imagine that if a diatom is a good competitor, it's good at basically excluding guys that it doesn't like. So the idea was to dig a bit into this and to ask, okay, who are the partners? Okay, fine, you exclude a lot of people, but what are they? What species are they? So when we started looking into what those organisms were, I quickly saw that most of those exclusions involved copepods. So diatoms exclude copepods that feed on diatom. It excluded dinophyces that can also compete for nutrients with diatoms. It also excluded polycysteines. Polycysteines are actually a class of microorganisms that also use silica to build their cell wall. And also it excluded Syndinialis that are a class of parasites. So it's a lot of stuff, you know, you exclude potential predators, potential competitors, potential parasites, and there's actually a very cool paper published in an obscure journal called Making Sense of the Ocean Biota from Viktor Smetacek, and he basically talks about the 3 Ps, predators, parasitoids, and pathogens, as being big drivers, like as being 3 types of organisms that exert selective forces on diatoms and drive the evolution. So again, shifting a bit from the nutrients or drivers of abundance to actually other organisms, predators, pathogens, parasitoids, are also exerting this pressure on diatoms to evolve. So he calls them the 3 Ps, and I thought, okay, do diatoms have a different behavior towards those 3 Ps? Are they really excluding them? Is it really different? Are they really special in that they are excluding those potential predators, or just every organism also excludes copepods, dinoflagellates, in which case there's nothing special about diatoms? So the way to address this was to start with Syndinialis. So this is a paper from Chambouvet et al. She shows a dinoflagellate that is full of this Syndinialis parasite. So you can see this is the cell. And within the cell, you have this kind of Christmas or Hanukkah, blah, 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 with the parasite that basically ends up exploding the cell. I think it's pretty harsh, but this is how it happens. And these organisms, those Syndinialis, are extremely connected in the Tara interactome. They are big, big drivers of all the interactions that we have. So here what you see, when I focus I took this time everything that involved those parasites. I wanted to see, do diatoms have a specific behavior regarding those parasites compared to other organisms? So here are all the different taxonomic groups that we have in the Tara Ocean interactome, and each time I look at to what extent they interact with those parasites. So here, on the right side, you have organisms that most of the time are positively correlated to Syndinialis, okay, so you find them with parasites. Whereas here on the left-hand side, you have organisms that we rarely find with parasites. Like if you look at the whole Tara data set, which is quite big, most of the time you don't see those organisms co-occurring with Syndinialis, with the parasites. And diatoms are right here. You can see that they are a bit in the middle, but you can easily do stats, binomial testing, and show that on average, they exclude those parasites much more than what you would expect at random. Actually, if it was all a random effect and that there was no difference between diatoms and other organisms, they would be somewhere around here. On average, they would tend. The fact is, there is, I cannot say an active process, but the fact is, on average, you see diatoms less with those parasites than what you would expect at random. And this is also true for potential predators. It's also true for potential competitors. So this was kind of addressing the 3 Ps of Viktor Smetacek. The thing is, you can also, once we define those, let's say, 4 major players, we decided to go a step further and to look at the topology of the network. So trying to see how the different subnetworks of the diatoms with those major partners did they differ? Maybe all those subnetworks are the same, maybe they all interact the same way, maybe not. So this time what we did is that from this big diatom network, we divided it in 4 major groups that are basically the 4 major partners of interaction with our diatoms. We have the copepods, we have the dinophyces, the marine straminopods, and the parasites. Each time in a network, you have in yellow the diatom node, in blue the partners, so all of these nodes are basically copepods, and the size, and from this network you can start doing graph topology basically. So typically the size here, the bigger it is a metric called betweenness centrality, and this can tell you to what extent your species is actually connected to everything else in the network. So here are basically pointing toward different keystone species. The thing is, the species that are here, big in copepod or big in dinophyces, are not the same. So you have, based on the major group of interaction, either if it's a predator, if it's a parasite, or if it's a pathogen, it's not the same keystone diatom species. They differ. You also have metrics of connectivity. Connectivity basically asks, does everyone interact with everyone? If I go back to the predator-prey typical case, it will be, does a copepod eat every type of diatom? Like, it doesn't care, it's a diatom, he will take the big one, the small one, the long one, or will they have a more specialist diet, right? Typically, connectivity will tell you that. It will look at each of your diatom nodes, it will look to how many different copepods it's connected, and same side on the copepod, it will look, does the copepod eat everything or not? And what you see is that for the parasites in the marine straminopyle, it was actually a much more specialist type of interaction. It was one specific diatom with one or two specific syndinyales or parasites. And on the contrary, for those predators and those dinophyces, it was much broader. It was much more generalist in the sense that, for example, the co-occurrence or the exclusions were much more spread throughout the whole group of diatoms and copepods. Finally, you can also look at correlation value, because it's one thing to look at those metrics, but each of the edges, right, the links that connect the two species also carry information on the strength, typically your strength of your exclusion. So I started looking at the difference of correlation values, typically with a predator or with a potential parasite, here, syndinyales. And what was interesting is that on average, the exclusion values here were much stronger for the parasites and the marine straminopyles than for the copepods and the dinophyces. So it meant somehow that here the correlation was stronger, so they tend to much more exclude those potential parasites than those actual copepods. And finally, also, one point was to try and see if we needed a control, a kind of control group, okay, because here everything is very based on the diatoms. So we took the polycysteines. It's a good control group in the sense that it also has silica. It's approximately same size range as diatoms, and we conducted exactly the same analysis on them. And basically what you show is that the pattern is much more random. So it also kind of encouraged us in the direction that diatoms were really good at competing. What I saw also rapidly from those results, and this was also with work from a former PhD student in Chris's lab, Shruti Malviya did all the work on characterizing diatom biogeography based on the Tara Ocean data. The names that I had here, all the names of diatoms that interacted, that were highly connected, didn't really match with how abundant they were in the ocean, which brought me to think, are the abundant diatoms actually important in structuring the community? There's always this idea that, if something is abundant, it's super important, but it didn't seem to be so true at this level. And so I could easily check that basically the most abundant diatoms based on the whole Tara Ocean's data weren't necessarily the most connected, and they weren't necessarily the most central. Which was a bit counterintuitive, but at the same time not necessarily not surprising, but this was something that you have to keep in mind. It's not because something is abundant that it's always the most important. And then when you dig basically at the genus level, we know we can, from the Tara data, we can differentiate different species. Okay, within one genera we can differentiate different species. And we wanted to see if there was something specific at the species level. Is it just all one specific genera that behaves the same? Or do you have something specific within each genera where only specific species will interact and not others? So here, this time, what you see is all the interactions that involve one specific diatom, which is called Chaetoceros. What you see are, on the one hand, the positive edges that include that Chaetoceros, and on the other hand, negative edges. And each of the columns here, are different groups. Here, for example, it's arthropods, dinophyces, and this is the total number of interactions. And what you directly see is that in terms of exclusions, this Chaetoceros excludes mainly syndinyales, marine straminopyles, dinophyces, arthropoda, so the same organisms that we talked previously. And on top of that, the different colors give you the different species. And what you see here is that most of the exclusions are driven by one single species. It's called Chaetoceros debilis. And on the other hand, in the positive correlations, it's driven by only this gray species, which is Chaetoceros rostratus. So it's not the same species. You have a species-specific pattern of exclusion and co-occurrences, and this is not only true for Chaetoceros, it's true for all of the other diatoms that are important in this whole network. And even though we don't have the proof, just from the literature, Chaetoceros debilis, it's known to produce aldehydes, it can damage fish gills, so it has this potential to be harmful towards other organisms. So is it something that you can go towards to and maybe think more about what type of mechanism is behind, but at least it was interesting to see that the specific species of Chaetoceros that actually excludes all those bugs is known in the literature to be slightly harmful. So you can go a step further and look at where this pattern comes from of interaction. So here, typically, there was a lot of interactions between this diatom and these copepods that you can see here. The copepod is a potential predator of this diatom. Here are all the different stations in the Tara Ocean. This is the abundance of the two partner. In red, the copepod, and in blue, the diatom. And the big colored blocks correspond to different ocean provinces. So here it's the Mediterranean Ocean, Red Sea, Indian Ocean, etc. This is what drives a correlation. Okay, this is the abundance pattern between a negative correlation. You have different types of scenarios, but this is how we get from abundance of two organisms to actual two nodes, right? What you see here is that the diatom in most of the stations is actually pretty low in terms of abundance compared to the copepod that is pretty high, except in two stations. Here in station 67, which is actually blooming station in South Africa that you mentioned previously, and also in this station, which is in the Southern Ocean, station 84. And what is so special with those stations? They are blooming stations, so we know that these are stations in which there's a high input of nutrients that triggers a diatom bloom. It's combined with decrease in abundance of this potential predator, and this typically is what drove many of the patterns of negative correlations that we had due to those blooming stations. So it makes sense. So we've been through a lot of, let's say, graphs and everything, but as you know, you don't know, but I'll tell you, one of the main bottlenecks that we have right now in this field is obviously that we can't really assign biological interactions to those co-presences or exclusions. I mean, there's a lot of things published, but not many people go to really try and go beyond just describing networks and really trying to infer actual biotic interactions on those correlations. And the same bottleneck was actually observed in, for example, protein-protein networks. And one of the lifts that they had to try and address that was to try and map empirical knowledge on those protein-protein interactions. So we decided to do the same and to try and see, okay, how much of those networks can we actually recover from the literature? You know, just at least to know what's true, what's potentially true, what's potentially unknown. So here, it's basically a representation of everything that we know from the literature regarding diatom interactions. I curated around 1,500 publications that are real publications. It's not correlations. It's people that did the study in the lab that characterize those interactions. Comes from a variety of different sources. And what is really interesting when you basically classify all those interactions is that around 60% of them are experiments of people that are feeding diatoms, like feeding predators with different diatoms. So 60% of what we know about diatom interactions is about predation. A lot of it also is about insects. We know that in freshwater we can find diatom sequences in insects. So this is also the type of stuff that, you know, we're not really interested in insects in the ocean. So, right, it's knowledge that we can't necessarily use. And also most of the diatoms that are studied are freshwater diatoms. In terms of diversity, it's freshwater. So it means that even in terms of actual knowledge that we have, a lot of it is not necessarily adapted to the marine environment. And again, what the real power of this, though, is that everything has been validated. It's actual science. So now the question was, can we overlap this on this huge network of 5,000 interactions that we have, and what can we recover? So when we map this empirical data on the network, this is basically the subnetwork from everything, all those correlations. If we try to extract everything that has been at least once described in the literature, we can recover around 20% of the literature. Like 20% of what we know from the literature, we find it in the Tara, which is a kind of true positive. But this represents mainly symbiosis, competition, and predation by copepod. There are a lot of things that are absent. Typically from the literature, there is a lot of worms-related interactions that we don't find in our network. Parasitism also, we have a hard time recovering parasitism in this technique. No bacteria. Typically in the literature, we have only, I think, 5% of the papers that concern interactions between diatom and bacteria, but diatom-bacteria interaction represent 20% of the correlation network. What does it mean? Is all of this unknown, or is it just potential directions in which we should go? But still, it shows, this previous picture just shows how skewed our knowledge is, in the sense that the diversity of marine diatoms is much higher than that of freshwater, and yet it's completely skewed in terms of knowledge that we have about them.
Flora?
Yeah. 5 minutes? Oh yeah, that's not good. No, but I started late.
It's okay.
Okay, I will accept.
We have also questions in the
But anyway, to make a long story short, there are many things. Typically, when we look at this big correlation network, even though we still can't validate everything, we do know that there are species that are highly connected and that don't even appear in the literature. So maybe these could point out towards new species that we can maybe focus on and try to cultivate to understand why they are so important in structuring the community. So to summarize this first part, I showed you that diatoms can act as segregators against 3 major P's that are the parasites, predators, and pathogens, that abiotic factors only drive 13% of the interaction, and most of it is actually biotic, that topologies and correlation can hint you towards the specificity or the generalism of species interaction, that it can also infer on the fact that abundant diatoms are not necessarily the most important in structuring the community, that those interactions are species-specific at a global level. It's not like all the genera behave the same way, but even within one genera, you have specificity, and that we also need this literature to be able to map it and to kind of confirm that what we are doing has biological meaning. So I will skip this because it's not the most interesting part, and move to the second one that I will have to do quite fast, which is much more like, if you don't like bioinformatics, this should be better. Typically, I spend the first part of the presentation saying to what extent there are good segregators, but we do have evidence that it's not always the case. So typically, these are confocal microscopy pictures taken by Sébastien Collin. Again, order of magnitude is maybe 20, 50 micron. All of these picture at least one diatom. Here it's a centric diatom, and all of these diatoms are in interaction. So here it's a diatom, centric diatom. It's surrounded by those small organisms that are nearly, it looks like a spacecraft with equidistantly situated small spheres. Here it's a diatom, a chain-forming diatom from the Arctic. You can see the chloroplast, what seems to be parasites inside. This is what we call corythron. It's also a diatom, and you can see here that it has those small organisms attached to it. We don't really know what's happening. Again, this is a Chaetoceros with Vorticella. You can see that it's attached here. This is also another Chaetoceros with those small, probably Phyllocystis attached to it. So we do have a lot of evidence that in many cases they do interact. But for most of those interactions, we don't know what's the genetics behind. We don't know, we can observe them, but we cannot really grow them. We don't know in what environmental conditions they occur. And because I was also a bit fed up with networks, I actually wanted to see stuff. And I decided to work on this specific interaction. What you see here is one diatom, this one. It has the shape of a banana. It's called Fragiloropsis. And you have to imagine that it's 5 or 6 diatoms that are stuck together. So when you stick the bananas together, it makes a kind of a barrel, okay? So this is the barrel. And inside you have this. It's a tintinid, also a unicellular organism. It's a heterotrophic protist. And it's sitting inside for the moment. We'll stop here. It's been described, I think, once in 1993, but it's like just one picture. We don't see anything. And what happened is that based on Sebastian's data, we knew that specifically in those stations in South of Africa, it was really abundant. Like in all the microscopy data, we have high throughput microscopy. It was really abundant, and we didn't know why, never characterized. And I said, okay, let's go, let's try and characterize this at a larger global scale, because there was the whole potential of the Tara data, in the sense that Tara, it's not only genomics. We have, for each and every station, for each and every size fraction, actual water that is still kept in the fridge and that you can go back to kind of confirm stuff that you observed a posteriori. So what I did is that I isolated those organisms from both of the partners, we managed to sequence part of the ribosomal RNA, specifically the V9 sequence. It's 130 base pair long. So for each of those organisms, I had 2 sequences, one for the diatom, one for the tintinid. And then from that, you can recover the abundance throughout the whole Tara dataset, because this is exactly the marker that we use to do the abundance of the microorganisms in the ocean. So I went back to the data, and from the sequences that I found here, I mapped the abundance of the diatom on one hand, of the tintinid on the other, and saw that, okay, the diatom is really abundant here. My tintinid seems to be abundant here. But then the question was, if the 2 organisms actually co-occur, if they are both abundant in the same station, do they actually interact? It wasn't necessarily obvious. So this is another way of looking at the data. These are the different size fractions that we sample. All of the stations and the different blocks that you see are again, ocean provinces, and the abundance of the 2 organisms are the different colors that you see. So what I did is I directly targeted stations. Here, this is the 20 to 180 micron fraction. I targeted stations in which the 2 sequences are abundant, and went back to the actual samples, looked at them under the microscope, and wanted to see if I could recover the interaction, which was pretty cool because it worked. That was nice. In a couple of maybe 15 stations, just looking under the microscope, you discover that instead of looking in 40,000 different samples, you can use the data to actually target what you're going to look for. And this is how I isolated a new consortia that then we wanted to characterize. So the first step was to try and characterize the morphological diversity of this consortia. So you can see here it's a different type of tintinid, so we wanted to go a bit deeper into that. What you see here is that there's a high morphological diversity, at least of the tintinid, but not of the diatom. The diatom is always the same. As you can see here, it can form sometimes just 5 to 6 chain, but sometimes the diatom chain can nearly enclose and form a whole barrel that completely encapsulates the tintinid that is inside. From scanning in color microscopy, we saw that there wasn't any proof of mechanical attachment, which kind of hinted us to the idea that it might be just some kind of exopolysaccharide that is combining the 2 organisms together. We also saw from the confocal microscopy that the diatom content here, the nucleus that you can see in all of the chains, was intact, which also was a bit different from all the previous interactions that we know, where in general with diatom and tintinids, the diatom is completely dead, whereas here it seemed that the diatoms were still in good shape. And most of all, the diatom was actually, this diatom is known to be the only one that can form this barrel shape. So to go a bit into depth, we isolated all of those different consortia, sequenced all of them, and tried to characterize them genetically. I'll try and go fast. What you see here is for the tintinid. It's called salpingella. From all of the different interactions that we isolated, we see that there are 2 major haplotypes, so it's not exactly the same type of tintinid. We know from the Fragileiopsis side that it branches very differently from all the other Fragileiopsis that we know. It's called Fragileiopsis doriotus. It's the first time we actually have sequences for this organism. And what is nice is that it's the only one also that is capable of forming this barrel. And we until now didn't really have genetic evidence that it was actually different from the other Fragileiopsis. When I wanted to go a step further, I don't know if you're familiar, but when we do high-throughput sequencing in general, we sequence all the ribosomal RNA that we have, and then we create species by combining together sequences that are 97% similar, and anything that is 97% similar, we say it's the same species. The problem here is that within the diatom, our sequence of our diatom was 99% similar to another sequence of another diatom that was completely different. And this really shows, if you are using those type of clustering techniques, that you have to be really careful when you do this 97 or even 98% clustering, that you are basically combining and putting together sequences that belong to organisms that don't have the same morphology, that don't establish the same interactions, that probably don't live in the same environments. So. And even worse, in terms of genetics, within one single diatom, we had different versions of the same sequence, which in terms of, there's a lot of inter and intra single cell diversity. Regarding haplotypes, one of our questions was when we saw that our tintinid were 2 different types of haplotypes, we thought maybe it's related to geography. So this is what we call a statistical parsimony network, where, for example, this is one sequence corresponding to one tintinid, and each of the edges is basically a base pair difference. So it's 3 base pair difference from this one. And the color corresponds to the station from which it's been isolated, that you can see here. So typically here, you can see that a lot of sequences were similar, which was haplotype 2. But what you can quickly see, for example, here from haplotype 1, so all the sequences here are strictly identical, but they come from very different places. One of them comes from Marquesas Island, from west coast of Peru, from South Africa. So there's no relationship between the genetics and the geography. On the other hand, for the diatoms, it was a bit different. We also saw variability within some of the genetic markers, and we saw that this haplotype, those sequences were actually quite similar. They all corresponded to one station in South Africa, which was a blooming station, which also showed that there was biogeography in the specific genetic traces. So then the last question was to try and see what was the ecological importance of the consortia. So how widely was it distributed? What did we know about the diatoms, etc.? I will go fast, sorry. First of all, one thing that was interesting is that in the Tara data, because there was no genetic information about this diatom, it was initially targeted, like assigned taxonomically as an unknown, because we didn't have it in any reference database. It was an unknown diatom. It turned out that it's the second most Fragilaropsis in the ocean. It's actually the 7th most abundant diatom in the Tara data. So it shows the idea that let's say the back and forth between the experiments, the morphology, the sequencing, and back to the genetics actually improved the assignment. So we managed to assign quite a bit of sequences thanks to that. We also know that in some cases, nearly 100% of your tintinids will be associated. It tells you that maybe in specific conditions it's an obligate interaction, and sometimes this interaction actually represents 50% of your total tintinid community. So what was interesting when we did a quick, like partially square analysis is that here are the different tintinid barcodes, and these are the diatoms. And what you see is that they're basically opposite in terms of environmental condition. So what this means is that they don't favor the same abiotic environments, but still you find them interacting together, which kind of hints again to the idea that biotic interactions might be a good driver of community structure. And so what was interesting is then, okay, if they don't share the same environment, maybe there's something with the species that are present. And what comes out from the analysis is that this interaction between the diatom and the tintinid actually occurs in stations in which there are a lot of potential predators, a lot of copepods, and a lot of heterotrophic other protists. What I mean is that the interaction is not obligate in the sense that we do see the diatom free living, we do see the tintinid free living, but stations in which 95 or 100% of the community is associated are actually stations that are blooming stations, that there's a lot of predatory pressure, so maybe defense mechanism. I can move, but we discovered tons of other stuff associated, which was very cool. And because it's a bit, that was a cherry on the cake, you can actually see a live video of what it really looks like. This was a collaboration with the people in Brazil. Again, I went to a seminar without really knowing anyone, and I just showed a picture of this to a Brazilian guy who told me, that's really funny, I see it every year. And I was like, what do you mean you see it every year? I see it dead for 3 years, so you mean it's actually living? So he sent me this video, which was kind of very cool for the reviewers also, because one of the major criticism we had was maybe all of your fixed interaction, your interaction is a product of a fixation, which is totally possible because we didn't have live material. So this was kind of the evidence that it really happened. So I'm sorry because I thought you didn't see the interesting part. Here you can see that, okay, it's moving forward. So this is the tintinid, right? You have the small cell with the ciliate, and it bumps into this dinoflagellate, Serratium, and it rewinds, and it goes away. And there are tons of video like this where you see that there's a sensing mechanism, which means that does this like serve as a shield, and then it's going away. I think it's really crazy. I have other videos if you like, but this in terms of behavior and dynamics was quite interesting to see. So from this, you see that, okay, data-driven approaches can help to drive decision and research direction. that we had high morphological diversity, that we also show and assign the first available sequences for that species, that more generally it was the first time that we could actually characterize molecularly an interaction at high spatial scale of a diatom symbiosis, that even though we had abiotic factors, they weren't enough to predict the community structure, and of course, burning issue, why? Why is this happening? Is the tintinid getting benefits from photosynthetic exudates? Is the diatom gaining motility? Diatoms don't swim. When they attach to a ciliate, they swim, they break boundary layers, they get access to new nutrients, they protect themselves from grazing because they are bigger, it's a kind of armor. Bacterial recruitment, increasing sinking rates, like hypotheses are endless, but I had to end my PhD. And most of all, you don't have them in culture, which is a big bottleneck. I think I will just go through this. To give you an idea, right now what we can do now with the data, with the type of data that we have, is to identify based on morphology, based on genomics. We can quantify. We have high throughput microscopy. It's not only about sequences, but we can also use metabarcoding still. We can functionally characterize. We can take the transcriptome of these interactions, recover their abundance and their expression in the whole ocean. We can contextualize. We can use those interactions and see in which environmental conditions they occur. And also we can look at host-associated microbiome. And all of this kind of opens broader perspectives to improve the spatial-temporal distribution models that are today very based on niche and neutral processes, but not always implementing interaction. We can also connect those interactions to global chemical cycles, look at coevolution between species, look at community structure, etc. Final thing, obviously, my 2 papers are a drop in the ocean of the Tara stuff. It's a big collaborative initiative, and most of all, the data is out there. If you want to know how species are distributed, how genes are expressed, where genes are present, in which species of the ocean, of the microbes, your genes are there, it's all there, and the goal is for everyone to use it. And I think some of the best papers that have been published come from last year, from people that are not related to Tara. Some crazy guy in Chicago just took all of the data, reassembled everything much better than what we did in the first place, and that's the whole point. Otherwise, it's this is how science works. So yeah, go ahead, and I'm happy to assist you. We also have, just for the curious between you, a huge platform called Ecotaxa, on which we have all the microscopy. All the microscopy is now available. You can see all the pictures of every organism in the ocean, and it's quite fascinating for coffee breaks. So yeah, thank you very much. I'd like to thank Chris, who was my supervisor, also Lucy Bittner and Shruti, who were the 2 my major Tara supervisors, and yeah, that's my old lab in Paris. Thank you.
One or 2 questions?
Yeah.
On the bioinformatics bit, first you showed that 87% of the interactions are not controlled by any abiotic factors.
Let's say, yeah.
Okay, we're not significantly associated with any abiotic factors. Did you also check there for combinations of different abiotic factors? You check whether a certain combination of temperature, nutrients, and that might bite a bit out of your 87%?
Yeah, you're probably right. Then you have so I didn't compute the network. Caroline Faust and Gypsy Mendes did it. You're dealing with a lot of data. So the combinatorial, if we start to do multi-environmental parameters and that you try to...
Okay.
So I think there was also a technical limitation in doing so, and already being able to control for one environmental factor for each of the interactions was quite a bit of a pain.
One other quick comment. You took your diatoms, you said they have far more negative correlations than they do positive correlations.
Not far more, like 100.
Okay, they tend more toward exclusion than cohabitation. And you said that this makes sense because this is a very competitive organism. Because you're dealing with correlations, it could also mean the exact opposite, that they're just very, very sensitive to all of these biotic effects. They're excluded from every place where there are predators. They're excluded from every place where there are pathogens. They're excluded from every place.
You mean rather than being good competitors, they are like victims?
they need very specific sets of conditions where they're more or less alone in whatever piece of ocean in order to thrive.
Yeah,
I think also makes sense with growing blooms.
You can see it both ways. So here, I guess the best way to do it would be to look at dynamics to see, like, are they preceding the demise of other predators, or is it the other way around? Here, one of the main drawbacks also of the data is that you're working with time points. Each time we went, they went in the station, it was a snapshot. So it's very hard, and it's true that it's something that we can't. I don't think we can really derive it from here. But in general, we know of defense mechanisms of diatoms, right? When they produce aldehydes and that they decrease egg hatching success in copepods, it's not like they can survive just because copepods are not there. They also actively...
whether they do it well.
Whether they, not so well apparently. But it's... I agree.
For the sake of time, we'll
Okay. Completely over
fantastic talk.
I'm
No, come on, fantastic talk. And naturally, I have many comments.
Yes, sure.
One of them is that it's a very general one. When you go in the ocean, and I think you probably know that, when you take samples, you just take a slot in time and space, and you can produce several times already in front of